Hello students, welcome to LEELA'S tutorial. So in today's class, we will discuss about development of Cybrids and their applications. Right? So here in last class, we have discussed about the protoplast, culture and fusion. Protoplast are the cells which are the plant cells devoid of cell wall. Right? Or simply you can say the plant cell without cell wall is referred as protoplast. So these protoplasts are fused so that you produce a new variety or a hybrid variety plants, right? So when two intact protoplasts are fused either by means of induced fusion or by means of spontaneous fusion, here the cytoplasm and the nucleus of two different plant species protoplast will fuse to produce a new plant variety, right? So here let us discuss today how these cybrids are produced. So here the cybrids are somewhat different from the hybrids, right? In case of hybrids, it is the two protoplasts which fuse so that their nucleus and the cytoplasm will get merged to produce a hybrid plant. Whereas in case of cybrids, only one cell or only one plant species protoplast consists of nucleus and cytoplasm, and other plant species protoplast consists of only the cytoplasm. So here there is only one nucleus in the cybrid plant and two different cytoplasms in that particular cybrid, right? So let us see in detail. So here the cybrids are produced by combination or fusion of non-nucleated cells to cytoplasm of nucleated cells. So what do you mean by non-nucleated? Non-nucleated cells are also referred as enucleated cells, or you may also call them as cytoplasts, right? So if the plant cell wall doesn't consist of cell wall but has all the cell organelles, then you call them as protoplast. Is that clear? So here it is, the cybrids are produced by combining the cytoplast cells with the protoplast cells. So here you are not going to take the same plant species, right? It is not the same intrageneric or intraspecific because their normal pollination will occur, right? Whereas here we are going to produce the cybrid plants or fuse the protoplast of the plants which are interspecific or intergeneric, right? Where there is no natural pollination or natural fertilization occurs, right? So by this method you can produce a novel plant varieties which are useful for the agriculture. So let us see the technique. First you are going to select the plant A and plant B. So I told that these protoplasts are not from the same plant or from the intrageneric or the intraspecific. You are going to take a plant samples from two different plant species, right? Or two plants belonging to different genera, right? So here I am taking protoplast A and protoplast B. That means first I have selected the plant A and isolated the protoplast, and then again I have selected the plant B and isolated the protoplast from that plant B. Is that clear? So now the protoplast is defined as the plant cell without cell wall. You can see here the rigid cell wall is absent, right? So here the protoplast consists of nucleus and the cytoplasm and all other cell organelles. And here even in plant B, the protoplast consists of all the cell organelles along with the nucleus, right? So if you are going to fuse both the protoplast of two different plant species, then you are going to produce a hybrid variety, right? Otherwise you can say a hybrid protoplast is produced when it is cultured in plant tissue culture, then you produce a hybrid plant variety, right? So here you are not going to fuse these two protoplasts, but here you are going to make some changes so that you are going to produce a cybrid plants. Okay, so what you are going to do in the protoplast A, you are not going to make any changes. Whereas in plant protoplast B, here you are removing the nucleus, right? The nucleus is removed out of the cell. Now this protoplast is called as non-nucleated cell or enucleated cell or cytoplast. So here you can see, after this is the protoplast. When you remove the nucleus, then you call it as enucleated protoplast or cytoplast or non-nucleated cell, right? So now this protoplast B will be fused with, this cytoplast, you can call it as cytoplast B, is fused with protoplast A. Why I am calling it as cytoplast? Because it lacks nucleus, right? But cytoplasm and all other organelles are present. Okay, so now fusion of protoplast A and the cytoplast B will result in cell fusion, right? So what is the involvement here? It is the PEG, that is polyethyl glycol. So what is the mode of fusion here? Is that an induced or it is an spontaneous? It is induced, right? It is an chemical induced method where you are making use of fusogen or fusion causing agent, that is polyethyl glycol. So once you use this in a beaker, right, add all the protoplast A after centrifugation. That means you are going to purify all the protoplast A cells and then B cells. Then you are going to enucleate the B cells. Then add all your enucleated B cells and the protoplast A cells, right? And then you are going to pour the sufficient amount of this polyethyl glycol and incubate them. So when you incubate them, this fusogen or fusion causing agent will allow your protoplast to get fused, right? So here, if the same protoplast A and A are getting fused, you are going to, again by using the marker genes, you are going to separate them. Okay, that is of no use. Again, this also is of no use without nucleus, right? So you want a hybrid or a cybrid protoplast which consists of the cytoplasm and the nucleus of the protoplast A or the plant A and only the cytoplasm of plant B, right? So here, why you are removing the nucleus? You want the characters present in plant A and B to be present in a new plant, right? So that you will produce a hybrid. So why you are producing a cybrid when you have the hybrid? So that is because here you have the beneficial characters in the cytoplasmic DNA. That means you want only the mitochondrial DNA to get inherited, but not the nuclear DNA. For that is the reason you are going to remove the nucleus. So once there is a cell fusion, gradually the membrane, the plasma membrane present in between both the cells will get dissolved and there is fusion of cytoplasm. That is referred as cytoplasmic fusion. So once the cytoplasm fuses, you can see the larger size cell, right, which consists of fused cytoplasm of both the cells A and B and nucleus of only A. Is that clear? So now it is having the genetic material of plant A and the mitochondrial DNA of plant B. Is that clear? So as a result, the hybrid or the cybrid plant, right, the protoplast which is formed by fusion of your non-nucleated cell to the cytoplasm of nucleated cell results in the formation of cybrid, where it consists of two parental cytoplasm and one parental nucleus. Is that clear? So here now you have the donor plants. These two are donors. So in the first donor plant, you want the nuclear DNA to get expressed. Whereas in the second one, the cytoplasmic donor, where you are removing the nucleus because you want only the mitochondrial DNA to be expressed. So now the cybrid plant, which consists of the nucleus of plant A and the mitochondria of both plant A and plant B. Is that clear? So as a result, you are going to produce a new plant or the novel plant variety which is referred as cybrid. Okay, so if you see in detail the applications, here you can transfer the cytoplasmic male sterility. If plant B is having the cytoplasmic male sterility, so which is a beneficial character in some of the plants, right? So here you doesn't want any nucleus, but you want this cytoplasmic male sterility which is present in the mitochondria. So in that case also you can make use of this technique in order to transfer the cytoplasmic male sterility in Cybrids. Then next one is antibiotic resistance. See here, in case of agriculture, we want the crop to be herbicide resistant and also the antibiotic resistant. So in case of agriculture fields, we make use of this technique to produce the cybrid plants which may be resistant to antibiotics and the herbicides. And mostly it is to overcome the sexual incompatibility. Right? So I told, in case of two different genera, right, the plants belonging to two different genera or two different plant species, there is no compatibility or there is no possibility of pollination. Right? So when there is no possibility of pollination also, you can fuse or you can produce a plant which is having both the characters. In last class we have discussed about pomato and the tomato. Right? So here in case of tomato and potato, you are producing a hybrid plant which is pomato. Right? But they are sexually incompatible. Natural pollination will not occur. Right? So here in the same way, in order to overcome the sexual incompatibility between the interspecific and intergeneric plant species, we make use of this technique, right, where you are going to produce the hybrids which are beneficial for the agriculture. Okay, if you have any doubts, put it in comments and subscribe for further videos.